Aging is an inevitable part of life, a process that every living organism experiences as time passes. From the moment we are born, our bodies begin a gradual journey of change, growth, and eventual decline. But why do we age? What causes our cells, tissues, and organs to deteriorate over time, and is there a way to slow down or even reverse this process? The science of aging, also known as biogerontology, seeks to answer these questions by exploring the biological, genetic, and environmental factors that contribute to the aging process. While aging is a complex and multifaceted phenomenon, scientists have identified several key theories and mechanisms that help explain why we age and how it affects our bodies. At its core, aging is the result of the accumulation of damage to our cells and tissues over time. This damage occurs at the molecular and cellular levels and is influenced by a combination of intrinsic, internal, and extrinsic, external, factors. Intrinsic factors include genetic predispositions and biological processes that are inherent to our bodies, while extrinsic factors encompass environmental influences such as diet, lifestyle, and exposure to toxins. Together, these factors contribute to the gradual decline in the function of our cells, tissues, and organs, leading to the physical and cognitive changes associated with aging. One of the most widely accepted theories of aging is the free radical theory, which was first proposed in the 1950s. According to this theory, aging is caused by the accumulation of damage from free radicals, which are highly reactive molecules produced during normal metabolic processes. Free radicals contain unpaired electrons, making them unstable and prone to reacting with other molecules in the body. When free radicals interact with cellular components such as DNA, proteins, and lipids, they can cause oxidative stress, leading to cellular damage and dysfunction. Over time, this damage accumulates, contributing to the aging process and the development of age-related diseases such as cancer, cardiovascular disease, and neurodegenerative disorders. While the free radical theory has been influential in aging research, it is not the only explanation for why we age. Another important concept is the telomere theory of aging, which focuses on the role of telomeres in cellular aging. Telomeres are protective caps at the ends of chromosomes that prevent DNA from unraveling or degrading. Each time a cell divides, its telomeres shorten slightly, eventually reaching a critical length that triggers cellular senescence, a state of permanent cell cycle arrest, or apoptosis, programmed cell death. This process acts as a biological clock, limiting the number of times a cell can divide and contributing to the aging of tissues and organs. Telomere shortening is influenced by both genetic and environmental factors, and researchers have found that maintaining telomere length through lifestyle changes or medical interventions may help slow the aging process. In addition to telomeres, another key player in the aging process is mitochondrial dysfunction. Mitochondria are the powerhouses of the cell, responsible for generating energy in the form of adenosine triphosphate, ATP. As we age, the efficiency of mitochondrial function declines, leading to reduced energy production and increased production of free radicals. This creates a vicious cycle, as mitochondrial damage further exacerbates oxidative stress and cellular dysfunction. Mitochondrial dysfunction is thought to contribute to many age-related conditions, including muscle weakness, cognitive decline, and metabolic disorders. Another important aspect of aging is the decline in the body's ability to repair and maintain itself. Our cells are equipped with sophisticated mechanisms for repairing DNA damage, clearing out damaged proteins, and maintaining cellular homeostasis. However, as we age, these repair mechanisms become less efficient, allowing damage to accumulate and impair cellular function. For example, the DNA damage theory of aging suggests that the accumulation of DNA damage over time is a major driver of aging. This damage can result from both internal processes, such as replication errors and oxidative stress, and external factors, such as exposure to ultraviolet radiation and environmental toxins. When DNA repair mechanisms fail to keep up with the rate of damage, it can lead to mutations, cellular dysfunction, and an increased risk of age-related diseases. The hormonal theory of aging also provides insight into why we age. Hormones play a crucial role in regulating growth, metabolism, reproduction, and other physiological processes. As we age, the production and regulation of hormones such as insulin, growth hormone, and sex hormones, e.g., estrogen and testosterone, decline, leading to changes in body composition, energy levels, and overall health. For example, the decline in growth hormone levels is associated with reduced muscle mass and strength, while the decrease in estrogen levels during menopause contributes to bone loss and increased risk of osteoporosis. Hormonal changes are a natural part of aging, but they can have significant effects on physical and mental well-being. In addition to these biological mechanisms, aging is also influenced by epigenetic changes, which are modifications to DNA that affect gene expression without altering the underlying genetic code. Epigenetic changes can be caused by environmental factors such as diet, stress, and exposure to toxins, and they play a key role in regulating cellular function and aging. Over time, these changes can lead to the activation or silencing of genes that contribute to aging and age-related diseases. Researchers are increasingly interested in understanding how epigenetic modifications can be reversed or modified to promote healthy aging. While the biological mechanisms of aging are complex, it is important to recognize that aging is not solely determined by genetics. Environmental and lifestyle factors also play a significant role in how we age. For example, a healthy diet rich in antioxidants can help reduce oxidative stress and slow the aging process, while a sedentary lifestyle and poor nutrition can accelerate it. Regular physical activity has been shown to improve mitochondrial function, maintain muscle mass, and reduce the risk of chronic diseases, all of which contribute to healthy aging. Similarly, managing stress, getting adequate sleep, and avoiding harmful habits such as smoking and excessive alcohol consumption can help promote longevity and well-being. The study of aging has also led to the discovery of certain genes and pathways that influence lifespan and healthspan, the period of life spent in good health. For example, the insulin/IGF-1 signaling pathway has been shown to play a key role in regulating aging in various organisms, from worms to mammals. Mutations that reduce the activity of this pathway have been found to extend lifespan and delay the onset of age-related diseases. Similarly, the sirtuin family of proteins, which are involved in regulating cellular metabolism and stress responses, has been linked to longevity and healthy aging. These discoveries have sparked interest in developing interventions that target these pathways to slow down or reverse the aging process. One such intervention is caloric restriction, which involves reducing calorie intake without malnutrition. Studies in various organisms, including yeast, worms, mice, and primates, have shown that caloric restriction can extend lifespan and improve healthspan by reducing oxidative stress, enhancing DNA repair, and promoting cellular maintenance. While the effects of caloric restriction in humans are still being studied, preliminary evidence suggests that it may have similar benefits, including improved metabolic health and reduced risk of age-related diseases. Another promising area of research is the development of senolytics, which are drugs designed to selectively eliminate senescent cells. Senescent cells are cells that have entered a state of permanent growth arrest and contribute to aging by secreting inflammatory molecules that damage surrounding tissues. By clearing out these cells, senolytics have the potential to delay aging and reduce the risk of age-related diseases. Early studies in mice have shown that senolytic treatments can improve healthspan and extend lifespan, and clinical trials are now underway to test their safety and efficacy in humans. In addition to these scientific advancements, the field of regenerative medicine holds promise for addressing the effects of aging. Stem cell therapy, for example, aims to repair or replace damaged tissues by harnessing the regenerative potential of stem cells. Researchers are exploring the use of stem cells to treat age-related conditions such as osteoarthritis, heart disease, and neurodegenerative disorders. While still in its early stages, regenerative medicine has the potential to revolutionize the way we approach aging and age-related diseases. Despite these exciting developments, it is important to approach the science of aging with a balanced perspective. While researchers are making significant progress in understanding the mechanisms of aging and developing interventions to promote healthy aging, there is no magic bullet that can stop or reverse the aging process entirely. Aging is a natural and inevitable part of life, and while we may be able to slow it down or mitigate its effects, we cannot eliminate it altogether. In conclusion, the science of aging is a complex and rapidly evolving field that seeks to understand why we age and how we can age more healthily. Aging is driven by a combination of genetic, biological, and environmental factors, including oxidative stress, telomere shortening, mitochondrial dysfunction, DNA damage, hormonal changes, and epigenetic modifications. While these processes contribute to the gradual decline in cellular and tissue function, they also offer potential targets for interventions that can promote longevity and improve health span. Advances in areas such as caloric restriction, senolytics, and regenerative medicine hold promise for addressing the effects of aging, but they also raise important ethical and societal questions. As we continue to explore the science of aging, it is essential to approach this research with caution, responsibility, and a commitment to improving the quality of life for people of all ages. Aging may be inevitable, but with a deeper understanding of its mechanisms and the development of innovative interventions, we can strive to age with grace, vitality, and dignity.